This then is 3.22, looking at microorganisms and making beer, yogurt and cheese. Again, these are the questions you could be asked, and now we're going to run through the answers. So, first off, describe the process of making beer. Okay, so you add multigrains to water, add hops for flavour, boil the liquid, cool the wort and run into a mash tun. Add yeast. Yeast is there so fermentation can begin, so the sugar gets changed into alcohol. Remove the yeast and then bottle the beer. Okay. Yogurt then, pasteurize the milk by heating it. Pasteurization is really important because it kills any bacteria. Add bacterial culture to cooled milk, leave at 46 for 4 hours, 46 degrees. Milk contains sugar called lactose. The bacteria changes the form from lactic acid. Sample, that should say not simple, sample to see if ready. The lactic acid will curdle the milk, lowers the pH, which helps the milk preserve the yogurt. Add colourings and flavouring, package the yogurt. Then finally, describe the process of making cheese. Add bacteria and rennet. The bacteria will again convert lactose into lactic acid. The milk curdles and separates into curd and whey. Rennet contains enzyme rennin, which changes the milk protein to curd. Drain off any of the whey, then the curd, milk solid, fats and proteins are pressed to make cheese solid. The cheese is left to mature to improve its flavour and consistency. Okay, name and define microorganisms. Microorganisms are organisms of microscopic size which may exist in single cell form or as a colony of cells. Examples include virus, fungi and bacteria. What is pasteurization? Pasteurization is the process that kills bacteria by heating to high temperatures and is used to treat milk, beer and fruit juices. Milk is pasteurized by heating the milk to a high temperature, 72 degrees for 15 seconds before cooling to 3 degrees. Define homogenation. Commercial milk is homogenized. This is done by pumping milk at high pressures through narrow tubes. This breaks fat globules into smaller droplets so that it stays as an emulsion. Fat does not separate out and flow on the top. Methods then, of preserving food, all different types here. Things that we need to do then to keep really good hygiene: wear protective clothing, make sure we use disinfectants, we wash up properly, removal of waste. Fermentation equation, so glucose plus yeast gives you ethanol and carbon dioxide. Equation for aerobic respiration then, glucose plus oxygen goes to carbon dioxide plus water. And then finally, describe the relationship between temperature and growth rate. Okay, bacteria growth rate will increase as temperature increases till a certain point. Okay, at which this is considered optimum. After that, growth rate will decrease.